So let's begin our fourth part in the patterns of inheritance lecture. Again, just trying to reemphasize, we've talked about Mendel and the genetic scenarios where you have a monohybrid versus a dihybrid and how we can use Punnett squares to make predictions about possible outcomes. In the last lecture, part 3, we talked about probability, about the rule of addition and the rule of multiplication. So these are things definitely to keep in mind and get comfortable with. This is how you predict the odds or determine the ratios of what you think is going to happen. Now, what do we think will happen in regards to these particular outcomes? So one way that this is applied, and I'm not encouraging this by any means, but I just want you guys to think about it. One way that the rule of multiplication, the rule of addition, and probability, and all this is applied is when people gamble. So if you guys saw the movie 21, or an older film with Matt Damon and Edward Norton called Rounders, they're basing their decision on, do I fold, do I take another card? It's all based on probability. What are the odds? The next card coming up is going to be a jack, or the next card coming up is going to be a 6. Is that next card going to push me over 21 if I'm playing blackjack? It's all based on probability. So if you're good with math, good with probability, and good with, all right, how many cards have been dealt and how many cards are still in the deck, you can make better decisions if you're playing poker or blackjack or roulette, things like that. Again, I'm not encouraging it, but just pointing out some of the best gamblers are good mathematicians who understand probability. Okay. So as we look at genetics, often we want to do a thing called a test cross. Now the test cross is how we can try to figure out the genotype of an individual who expresses a dominant phenotype. So if they appear purple, do you know if they're homozygous purple or heterozygous purple? Looking at them, you cannot tell. So test crosses are used to try to determine this. So the way we would do a test cross, you take your purple parent and you say, you know, they could be this homozygous big P or heterozygous big P little p. And we're going to breed them with a recessive individual, the white individual. Now we know white, because it's recessive, always is expressed with two little p's, two little alleles, two recessive alleles to express that recessive trait. So anytime a purple individual is bred with a white individual, the white will always pass on the little p. So if you take purple and white and they produce offspring, if all of the offspring are purple, we can feel pretty confident that that parent, the purple parent, was homozygous. Now, you don't want to base this decision on just 2 kids. You want to breed these two and let them have hundreds or thousands of kids. You need a sizable sample. Sample size is critical in genetics. So I would not feel comfortable saying, well, I think that this parent is homozygous purple if you have 4 purple kids. That's a very, very weak sample size. There's no validity in most people's minds when you only have a sample size of 4. Not going to trust that. If you were to breed those 2 parents together, purple and white, and you got 4 purple kids, and all of a sudden you're like, wait a minute. Or I'm sorry, if you have 140 purple kids, now we have a strong sample size. Then I'm going to feel a little more confident that, yes, this parent is homozygous. The same thing would apply over here. If you have a purple parent and a white parent and you produce purple and white, Then you say, okay, I feel that that parent is heterozygous. Now the sample size over here doesn't matter. If you, well, I shouldn't say that. You need at least 2 kids. But if you had 2 kids, one purple and one white, you can feel confident that the purple parent is heterozygous. And the reason why we can feel confident that they are heterozygous is the fact the only way you will produce a purple child from this parent and this parent is if the purple parent carries a recessive allele, if they carry that little p. So one white child means that little p came from the purple parent. You will not get both of the little p's from the white parent. It does not work that way. So when we start applying this to our family tree, please don't ever say, well, I have blonde hair, which is recessive, because my mom gave it to me. No, she did not give you your blonde hair. She gave you one piece of information. Dad gave you the other. And if dad has black hair or brown hair, a dominant hair color, and you're blonde, it means dad is a carrier. And you inherited his recessive allele, mom's recessive allele. That's why you express. So test crosses are incredibly important to help us try to determine the genotypes of the parents, especially the parent with the dominant variation, where they express a dominant variation. So Mendel helped establish this. This poor Austrian monk back in the 1860s spent years and years and years collecting data, sorting peas, crossing, breeding, keeping track of it. He was one of the first people to apply statistics to his science, to this. And he was looking at sample sizes of thousands and thousands of peas to come up with these ideas, this rules of probability, the patterns or principles of segregation and independent assortment. And in 1865, Gregor Mendel goes to the scientific community called the Linnaean Society and presents his research. And unfortunately, he got ignored. So this poor man spent years and years and years of his life researching this, establishing these foundations, and the scientific community ignored him. What I want you guys to do is think about what was happening in the world in the 1860s that would have somewhat detracted or diverted the scientific community's attention away from what Mendel was doing. So unfortunately, he was ignored. It wasn't until the early 1900s where his work was rediscovered by 3 different scientists. They were independently researching genetics, and they came to the same conclusions. They looked at Mendel's work from 1860s, and it matched up, the results matched up with him, and that's when we say genetics was officially born as a field of science in the early 1900s. So in the big picture, genetics is a fairly young scientific field. I know it's 115, 116 years ago, but that still is fairly young compared to things like anatomy and some other fields of science that have been around for thousands of years. This is why we're still learning vast amounts of things about genetics. There's so many things we just don't know yet. Great opportunities in this field for people to start to continue the research that's being done and advance it to the next level. So if this interests you guys, some great opportunities here in genetics. So Mendel established our foundations. Since then, we have discovered what are called extensions to Mendel. They don't fit his basic rules. There are some basic genetics that follow monohybrid, dihybrid scenarios, but a lot of our genetics, we're realizing they just don't follow the rules. Things like sickle cell anemia. those red blood cells, the shape of it. Skin color, great example. Look at the spectrum of skin color there. These are all humans. These are all people. Yet there's this vast degree of variation within human skin color. That's not a monohybrid cross. That is not a dihybrid cross. These are things we'll start talking about with these extensions to Mendel's work here. So the first one we want to explore is this thing called polygenic inheritance. Okay? Polygenic means lots of genes. Multiple genes control the physical expression or the phenotype. So polygenic inheritance, multiple genes control the phenotype. It's not one gene or one allele or one set of alleles that determines this. It's going to be a bunch of them. So when you look at the spectrum of height, and that's what this very old black and white image is showing us. Here's a spectrum of height in a senior class at a high school. That's controlled by a bunch of genes. There's no one gene that says you're 6 feet tall, or 6 feet 1, or 6 feet 2, or 5 feet 8, or 5 feet 7, or 5 foot 6, and so on. It's not one single gene. It's the combination of multiple genes coming together to create a particular variation on that trait. So when you look at a trait and there's this continuous variation, that often gives us the indication it's polygenic inheritance. Okay? So the example I use, one way to relate it is it takes the combination of multiple genes to actually get that to express. Think about old combination locks where you spin the dial 27, 14, 3. That specific combination causes the lock to open or the trait to be expressed. If you went 27, 14, 8, it's a different variation. It's a different expression. So that's the idea behind polygenic. Multiple genes control the phenotype. So we're learning certain traits are polygenic, while other traits are what we call pleiotropic or pleiotropy. Now, pleiotropy is when you have one allele that will influence multiple phenotypes. So one gene, one piece of DNA, one gene, big A, or big A, or little a, and that one allele impacts multiple phenotypes and physical characteristics. So our example here is cystic fibrosis. Okay? Cystic fibrosis is determined by one gene. If you inherit this gene, the gene changes the shape of a protein channel in your cell membrane. It's called the CFTR channel. It's down here is this little protein structure. Now here's our normal channel. Here's the mutated channel. And the normal channel allows chloride ions to diffuse out through the cell membrane and outside of the cell, causing mucus and bacteria and things like that to literally just kind of get washed away from the cell. If you have a mutation of that single gene, that one allele, and your CFTR channel is not correct because it's built differently, chloride ions do not diffuse out. This causes mucus to build up. This causes sinus problems. The lungs get built up with bacterial infections. They get thickened because of the mucus. Sweat glands produce salty sweat. All of these different physical traits, these are all phenotypes, are the result of that one gene being mutated. Okay, so that is the idea behind pleiotropy. One gene influences multiple traits. When we look at polygenic, we have one trait that is influenced by multiple genes. So they're kind of an opposite to each other. So funnel, think about a funnel. Here you have the neck of the funnel is the gene, and then it opens very, very wide to create multiple traits. In that polygenic inheritance, you have a bunch of genes, very wide spectrum of genes that all narrow down into that one variation on that trait. Okay, so polygenic, pleiotropic, no criticism to Mendel, but things he didn't see. He figured out the foundation. Without that foundation, we wouldn't understand polygenic and pleiotropic inheritance.